Sure.
Okay.
I think let's start.
Is the recording going? Oh yeah, it is. So I've been asked to introduce today's speaker, so I'll do that. As I assume everybody knows, today's speaker is in the upper left-hand corner of my monitor, Wolfgang Stein. Wolfgang arrived to our world from the land of stick insects in Germany, where he studied sensory motor integration. He was a postdoc in my lab in the late 90s, during which he met the crab STNS and studied various aspects of co-transmission, primarily involving MCN1 and the IVN neurons. I still remember that his screensaver during those years was the streaming set of symbols on a green background that represented The Matrix, because that movie had just been released when he was in my lab. You don't still use that, do you, Wolfgang?
No, but it's still green, but not like, scrolling down.
So from my lab, he returned to Germany to a 10-year faculty position, 10/year faculty position at the University of Ulm, where he primarily returned to his first focus of sensory motor integration, but this time in the crab somatogastric nervous system, from which he and his group have generated a number of lovely papers. Following Ulm, Wolfgang came back to the States, moving to Illinois State University in the interestingly named town of Normal, Illinois, where he has thrived and is now professor of neurophysiology in the Department of Biological Sciences. Many of his former trainees continue to apply their trade in neuroscience, including one of our co-hosts for these monthly meetings. Right, Nellie? One highlight of my periodic collaboration with Wolfgang, at least for me, was actually a consequence of his transition from Ulm to Illinois, because it gave me the opportunity to host one of his late-stage students, Florian Diehl, during the time of the move. And while Florian was in my lab, that collaboration generated a gratifying paper where we were able to show that different gastric mill rhythms not only occur in the dish, but also occur in the intact crab. Wolfgang has actually just returned again from Germany, where he was doing a sabbatical. And hopefully that experience is going to be a major part of the story we're about to hear. So without further blather from me, here's Professor Stein.
Thanks, Mikey. I think the sabbatic is actually not going to be much of it. It's just a very short part at the end.
Oh, fine.
Sorry. Okay, let me share my screen. So, Mikey, it's still green. See, it's not.
Excellent.
But no more Matrix. I haven't even seen the new movie, frankly. It's on my list, but I haven't.
Yeah, it's not that great.
It's not that great. Oh, good. I mean, bad, but okay. All right. So let me talk a little bit about, I'm going to give you a little bit of a historical perspective. So where I come from. So I'm going to go back to stick insects for a little while, and then talk about the stuff that we have done over the years. And then at the end, I'm going to get to the more current things as well. So generally, sort of I'm interested in, how sensory motor processing works. This is what Mikey already mentioned, right? And so, of course, there's a very simple schematic here where you see the central nervous system that activates the motor system. Then there's behavior and sensory pathways inform the central nervous system. Of course, we know this is very simplified, and there's a much more complex schematic that I can show you. And we already know that this is not sufficient. And we understand that the processing of sensory information is one of the most difficult tasks that the nervous system actually does. You can, on top of that, of course, add all the neuromodulation here. So, but at the beginning, I came in and I was interested in partly physics, partly neuroscience, and so the questions that I came in with were how is sensory information processed? How can motor output and behavior be coordinated and maintained despite the manifold inputs and perturbations that the nervous system experiences in its everyday life? And so, this will take us towards convergence and divergence of information, modulation of responses to inputs, and then, of course, feedback to neurons and circuits. And appropriately, I started out in a lab that worked on related questions. That was the lab of Ulrich Bessler, who worked on behavioral control, a little bit of cybernetics, but also neuroscience. And I brought a couple of photos because they find them quite interesting. So this is my former PhD advisor. That's my PhD advisor, not former. He's still my PhD advisor. And on his microscope, he was such a perfect person to dissect things. He had super fine skills, like motor skills. I was so impressed when I met him. I have evidence that he got a PhD because that's what happened to you when you get a PhD in Germany. You're being carried around by your colleagues. So that was interesting. But he was also very good at drawing. And so in the background, you actually see all the advisees that he had over the years. And I'm somewhere here in the middle, pulling the big cart, like the big, and moving the research enterprise forward. So I met a couple of interesting people there, like Holk Kruse, who was a postdoc advisor to me a little later. I met Ansgar Büschkes. I'm not sure if he's in the audience here. There he is on some retreat that the lab did. I met Nino Ramirez for the first time. Nino is not here today. He emailed me earlier. Has a different meeting. But I remember meeting him. He was lecturing a neuropharmacology course, and that was actually my first time dissecting a nervous system with Nino, the very first time. And the two things that stick out to me as in my memory was like, he served us tea in the lab, and I accepted the tea and I hated it. It was horrible. It was a fruity tea, and I could not have it. But it stuck in my mind. Like until this day, I'm like, this was a horrible tea. The other thing is that he was so patient with us because I went through like 4 or 5 locusts at the time, trying to dissect the nervous system, and I couldn't get it done. And he kept encouraging us to move on. And finally, I think I at some point managed to dissect the ganglion. Jorge was there at the same time, approximately when I was there. He was in a neighboring lab, not in our lab, but in the lab of Dr. Deitmer at the time. And I unfortunately didn't meet him, I think, when he was there. But I met other people like here, Pearson was sitting here. And so it was certainly an interesting time. Anyways, we worked with these critters. And if you can't see them, that's because they're really good at hiding. There's a walking stick somewhere in the middle to make it a little more clearer. So I have one on my hand over here. And what they are good at is posture control. And they are so good at it that they essentially don't move. That's an active process. They don't move and they hide very well. And at the time, a lot of interest was going into like, how can they control posture, but also how can they switch from standing to walking behavior if you're doing a lot of posture control. So I'm not going to talk much about this for time reasons, but I'm going to skip forward a little bit and ask sort of a sensory question that, I worked with a colleague of mine from Bielefeld University, Josef Schmitz here, standing next to Jessica Ausborn, a former PhD student of mine in lovely San Diego. That's the view out of our hotel room at the time at the Society for Neuroscience meeting. You can imagine it was a beautiful hotel. Anyways, we asked, where do sensory pathways converge? Do they converge? And if so, where does this happen in this whole big schematic of sensory feedback? And so what we knew at the time, we were working at this pro and retraction control of the legs. And we knew at the time that there were motor neurons that carry out that behavior. We also knew that there were excitatory and inhibitory interneurons. Some were spiking neurons, some were non-spiking neurons. And on top of that, we knew that there were at least 3 sensory systems that somehow influenced that prone retraction of the leg. Well, there were tibial movement proprioceptors, coxal movement receptors, and leg load receptors. So those are on different areas on the leg. But we knew they were all influencing this behavior, but we had no idea where this information comes together. And so this was one of the interests that we had. And so we did one of these crazy preparations where, this is the animal, essentially the head would be to the left, the tail would be to the right. These are the 2 legs. Do you guys see my cursor? Okay, these are the two legs. There's the ganglion in the middle, and we have an intracellular recording electrode inside of a sensory neuron and the axon of a sensory neuron. Those were blind recordings, so we couldn't see the axons. We just had to find them, like poke around and find them. We had a stimulus up here, one sensory stimulus, a second sensory stimulus out here, and the third one out in the leg. And so with these experiments, we then stimulated recorded neurons. This is a sensory neuron. When you stimulate its own sense organ, doesn't matter what type it is here, Importantly, this neuron responds really nicely with spikes to the stimulus down here. The spikes are truncated because they are like 100 millivolt large. And so you only see like the first few millivolts here. So you see there's nice spike activity. However, more interestingly, if you stimulate the other two sensory inputs that I've mentioned, and those are the ones down here, campaniform sensilla and other hair field, you see that these neurons don't respond with spike activity. But what you see is those depolarizations that occur in the membrane potential whenever you stimulate the other sensory neurons. And in fact, those, they don't lead to spikes. However, if you change the membrane potential of the sensory neurons, you can see that you can reverse those inputs. If you go more depolarized in the membrane potential, you see hyperpolarizations instead of depolarizations. And you can, of course, measure the reversal potential. And it turned out that those inputs reversed at about -60 millivolts, which was a good indication that those were due to chloride channels. And in fact, we could block them with picotoxin. So we're pretty sure those were chloride channels. Now, what did they do? Whenever they were activated, they diminished the amplitude of the action potentials and the input resistance in those sensory axons dropped. And so this was a very nice example of a presynaptic inhibition that we identified here that manifested itself through a primary afferent depolarization. The main effect here is that you're diminishing the output of that sensory neuron. The amplitude of the action potential goes down. You're reducing transmitter release. But more importantly, this was the first time that we actually were able to show, I think, generally among people, that there was a direct interaction on the level of the sensory neurons, that one sensory modality was affecting the output of another sensory neuron directly through presynaptic inhibition. So there's a convergence of sensory information already on the level of the sensory neurons before they actually contact the interneurons in the central nervous system. All right, we also recorded from motor neurons. We could identify postsynaptic potentials, excitatory and inhibitory ones. Long story short, we were able to identify lots of places of convergence in this little circuit that we are studying. There are the sensory neurons on the left. We saw these interactions between the sensory neurons. We saw convergence on the level of interneurons and on the motor neurons. So what does this tell us? There's convergence that exists on all levels of sensory processing, from sensory neurons to interneurons to motor neurons. And so in this little schematic, that would be up here, the red arrows here, and then down here towards the central nervous system, interneurons, motor neurons. So the thing that we didn't know about back then, or didn't care about, I guess, was that, of course, on top of all of these sensory motor loops, there are those decision-making neurons, modulatory neurons, right, that project to those motor systems and then make a decision on what's going on, like what motor pattern to activate. In the stomatogastриc nervous system, you can think of them as the descending projection neurons. And in fact, that's what we then moved on to. Like later on, we came back to this topic of convergence of sensory information when we looked at projection neurons. And so, of course, Mikey and others had already identified a set of projection neurons in the commissure ganglia, and here the names MCN1, 5, 7, and CPN2. And we had already known that there is convergence of sensory information in them. So we knew, of course, from the work of Dawn and Mark Beenhacker that mechanoreceptors, the VCNs, and the muscle stretch receptor GPRs, that they converge onto that set of projection neurons. And so we knew there was some convergence. Now, in my lab, we added to this, we studied the interactions of two more sensory pathways, a muscle tendon organ called AGR. There was work of Carmen Smarandaka-Wellman, who is now in Cologne on parental leave currently, and of a chemosensory pathway that descends from the brain called the IV neurons. And this was done by Uli Hedrick here. I visited her last year. She has her own lab on epilepsy in Tübingen these days. So what did they do? Well, they activated those pathways and recorded from these neurons. I'm only going to show you a few of those recordings. Here is a recording of MCN1 in the commissure ganglion. Then Carmen stimulated the AGR neuron. And what you can see is that there's a strong inhibition of MCN1 activity. On the flip side, this is data from Uli. When you stimulate the IV neurons and you record the MCN1 neuron, you see that MCN1 is excited by the stimulus. And so with lots of recordings, lots of blockers, lots of low calcium experiments, and so forth, we were able to come up with a schematic that looks approximately like this. And so AGR and the IV neurons both innervate all of these 4 neurons in different ways. For example, there is a chemical inhibitory synapse on MCN1, but a polysynaptic excitation that comes from the IV neurons over here. So main point on top of that, of course, the convergence from the other 2 sensory pathways, the VCNs and the muscle stretch receptors. We knew that there was strong convergence on the level of these modulatory projection neurons, right? They integrate sensor information from different sense organ, even different sensor modalities. Now, we try to get at the idea of like, is this functionally or behaviorally relevant? This, these different responses they have in the convergence that they show. And so we took a very sort of reductionistic approach to this. We looked at HER and we looked at the IV neurons and looked at what are the differences in the activation of the projection neurons here. And if you just take 3 neurons, MCN1, CPN2, MCN5, and then look at the 2 neurons, you see that MCN1 is inhibited by HER, but CPN2 and MCN5 are excited. On the IV neuron side, MCN1, all 3 of them are excited essentially. And so we were kind of wondering what happens if you now remove the MCN1 inhibition and make all of them look like they were activated by the IV neurons. And so the question that we asked was, do the motor patterns that are elicited by these sensory pathways actually become more similar? Because that's what you would expect if any of this matters, right? And so this was some very interesting experiments that were actually carried out by Uli again. And so just to orient you, this is the gastric mirror rhythm that you get when you stimulate the HER neuron. This is the gastric mirror rhythm when you stimulate the IV neurons. On first glance, they look very similar, but if you look at the LG neuron here in the middle, you can see that the bursts have different durations, for example. Also, when the DG neuron on the bottom part becomes active in a cycle, it's different between the 2. And there are also some differences in the GM neurons. And so to put this in like for the SDG people in a phase diagram, you can plot at when these neurons are active in a cycle of the rhythmic activity, and this is shown down here from 0 to 1. The blue ones are the IV neuron. As the IV neuron gastric mirror rhythm, you see the GM neurons up here. You see the DG neuron here and the LG neuron here. So LG and GM are approximately at the same time. DG is alternating with them. When you then compare this to the AGR stimulation, you see some significant differences. For example, the GM burst starts earlier, the LG burst ends a little earlier, and the DG burst a little later. And so the question was now, if we now replace the inhibition, right, in MC1, do they become more similar? So this is what we get. We were a little frustrated with this because there are some more similarities, but not fully. Like, for example, the GM neurons are no longer different between the HER stimulation and the IV neuron stimulation. The DG burst moved a little forward, but not far enough. And the LG burst didn't care. So the rhythms became kind of more similar, but also not as much as hoped. And the conclusion kind of was that the combination of active projection neuron activities may contribute to motor pattern selection. And of course, we are fully aware that we made a lot of assumptions in these experiments, right? We also probably ignored a lot of different neurons that are activated by our sensory pathways, plus the direct effects on the sensory motor system or the motor systems themselves. And so let me quickly talk about the other neurons that are activated by this. And so the projection neurons sit in the commissural ganglia. This is one of them shown on the left side. And there are a lot of neurons in there. When we counted, we actually didn't count 500 like in the literature. We found about 200 or a little over 200 neurons in there. But at the same time, we didn't know how many are actually involved in those sensory responses, right? We could locate approximately where the ones are that project towards, the SDG. They are located in sort of the medial posterior area of the COG. But more importantly, what we did is we used voltage-sensitive dyes to measure a large number of these cells while we stimulated sensory pathways to see how many of these neurons are actually involved in those sensory responses. And so in this case, we used the IV neurons again. So they are up here. We stimulated the IV neurons. We looked at the response of the COG neurons with optical recordings. At the same time, we stimulated the VCN. So this is the mechanosensory pathway that Mikey had worked on before, and looked at, again, how many are activated, right? When you do this, you get a picture like this on the left side. So this is a staining with a voltage-sensitive dye. You cannot stain all 200 neurons. It's not possible because it's a three-dimensional structure, but you can stain about 50 or so at a time and then see what their responses are. And so when you just do a single pulse of sensory stimulation, you'll find neurons that respond specifically to the VCNs, but not to IV. You will find some that respond to IV neuron stimulation, but not to VCN, and you will find some that respond to both. And in fact, if you go ahead and then stimulate for longer, not just a single pulse, but do a more physiological activity of those sensory neurons, you find that a lot of neurons actually respond to your stimulus. Think of those as mostly polysynaptic responses, but of course, with these dyes, it's not entirely possible to separate monosynaptic from polysynaptic ones. The fact is that if you stimulate for longer, you're getting a large recruitment of neurons. And overall, we found 98% of the image neurons that we saw responded in some fashion to both stimuli. And that suggested us that there's a large network that processes the stimuli in the commissure ganglia. And in fact, if you do a coherence map and via a Pearson correlation, that map just essentially tells you how related are the activities of the different neurons to one another. Left is the IV neuron stimulation, on the right is the VCN stimulation. You see a lot of red here, meaning a lot of these neurons show similar activities. You can, from that, build a functional connectivity map, and this is the bottom here. This is just really how related those neurons are in their activities. And if you take any of this, it tells you that there's considerable parallel processing of sensor information in all of these neurons. They all respond to your sensor inputs. Not just that, they actually, most of them are multimodal. So in that case, what you see here is neurons that respond to both stimuli are in orange. Neurons that only respond to one of the two stimuli is in blue, and then the black ones are the ones that didn't respond to anything. And so about 80% of all of our neurons responded to both stimuli, to both sensory inputs. And just keep in mind that we only tested two modalities here, right? We didn't even test like the AGR, for example, or some other sensor input, GPRs. So it's likely that more of them are multimodal and not just unimodal. So again, many neurons processing the sensor information at command. And in fact, there were some interesting findings too, that some of the neurons that responded, like with an excitation, for example, to an IV neuron stimulation, switched the sign when they responded to a VCN stimulation. So for example, to being inhibited. I'm going to point out the bottom left here. Those neurons, those are all cell bodies that are marked here. Like if they're yellow, they're excited. If they're blue, they're inhibited. This is IV neuron stimulation. See those three, for example, down here, they are excited by IV neuron stimulation, and they are blue on the right side, meaning during VCN stimulation, they are inhibited. So these results together are consistent with what we would call a combinatorial code, how information is represented in the COGs, less so with the dedicated pathways, because you would see only one set of neurons for one pathway, essentially, in the COGs. So, let's take a step back again and go back to our little schematic here. So the projection neurons integrate sensor information from different sense organs. There's a larger network in the COGs that are not necessarily projection neurons, but there is a lot of convergence of sensor information on that level. So, but there's also, there was a lot of convergence. There's also a divergence story here, and this is something that Mikey mentioned a little earlier. And so here's one of the famous figures from Mikey's 2002 Nature paper, where he had shown that projection neurons contain more than one transmitter. And when I started in the lab, that was something that actually blew my mind, because I had never considered this before. No one had told me, and I had clearly not read the right papers at the time. So here's MCM1 with, its 3 transmitters, 2 peptides, 1 classical transmitter, GABA, right? And so we asked about, do all of these transmitters target the same locations, right? Are they targeting the same neurons? Is there convergence of these transmitters, or is there divergence? Now, let me stop before I go to this. Let me start with the IV neurons again, because there's a story of divergence in there as well. And so these IV neurons, they sit actually in the brain of the animal. They project from the brain down to the commissural ganglia via a very complicated way, 2 ways essentially to each commissural ganglia. And then they also project to the stomatogastric ganglion. And so if you look, you can actually find that they have multiple transmitters. At the times we knew of 2 transmitters. One was a formurphamide type transmitter, peptide, and the other one was histamine, so an amine. And I know by now, I don't know what the latest is for the crab, but we know that in other species, like myosuppressin is the actual transmitter in the IV neurons. But what do we know about these IV neurons, and why do we care? And so here comes Uli again, and she was in a very heroic attempt. She recorded these neurons actually in an intact animal and looked at how they are activated and what they do. And so she did this, like I said, in intact animals. She tried optical stimulation, olfactory or chemosensory, I should say, stimulation, and mechanosensory stimulation. And guess what? Only the chemosensory worked, activated these neurons, and this is what you get. So she recorded the IV neurons on the IVN nerve. This is what you see here. It's an intact animal, right? This is not an isolated nervous system. And she recorded the motor activity, and she did a stimulation, chemosensory stimulation to the antennae, where she applied a food stimulus, essentially. And what you see is that you get rhythmic activity in the motor patterns. This is a gastric mill motor pattern that's starting after you apply that stimulus. And so she did some spike sorting and found out that the IV neurons are rhythmically activated with these chemosensory stimulation in an intact animal. And so this gave us a good handle on like how to stimulate in an isolated preparation to mimic that activity. And when you do that, and this is now from an earlier paper actually, where we happened to do the right thing, from Andy Christie and myself and Mikey, where we stimulated the IV neurons, and what you see is it produces a nice gastric mill rhythm that's also long-lasting. More importantly, that gastric mill rhythm here depends on the activity of the 2 projection neurons, MC1 and CPN2. MC1 you see up here on the iron recording, the smaller unit here, that's long-lasting activity or a long burst of activity whenever the LG neuron is on. Now, also importantly, we know that this type of activity can be mimicked by formurphamide application to the CUGs. This has been long known. This was Jim Wyman's work at the end of the 80s, I believe, early 90s, where we knew that when you apply formurphamide to the CUGs, you can sort of get that type of gastric nerve rhythm. Now, at the same time, we found that stimulating the IV neurons inhibits the pyloric rhythm, at least when you stimulate it for a short time. It does. And this is a recording of the PD neurons, the pyloric dilator neurons. And you see when you stimulate the IVN, you get an inhibition of the pyloric rhythm when you do that. And so, this can be nicely mimicked by applying histamine to the stomatogastric ganglion. And we also found that you can block this inhibitory response by applying histamine H2 receptor antagonists. So what does that mean? What do we think is going on here? So what we think is that we have formurphamide or the, whatever that particular formurphamide-like peptide is, and histamine. The formurphamide is then released onto the COGs where it activates projection neurons, and those projection neurons start a gastric mill rhythm. And the histamine inhibits the pyloric rhythm in the STG itself. We did not find any actions of histamine in the COGs at all. And so this suggests that there's a divergence of the co-transmitter actions so that peptides are being used in the COGs and the amine is used in the stomatogastric ganglion. And so over here, you see this again. Here's the brain with the IV neurons. We could not find actions on the COGs, but we know that MC1 and CPN2 are activated, likely by the formurphamide. In the STG, the pyloric circuit is inhibited by histamine, likely no effect by formurphamide. We're not entirely sure about this though. And the gastric mill rhythm, same thing. We know it's inhibited by histamine. There might be an action of fumirfamide on some of the neurons. So clearly some divergence of co-transmitter actions. And so Mikey was, of course, right when he suggested that. And so this was a great project. But then, over the next couple of years, he was like, why don't we look at MCN1 and make sure that we find divergence there as well. And so we did that. We went to MCN1, looked at captor, proctin, and GABA responses in the STG. MCN1 again innervates most of the neurons in the stomatogastric ganglion. Here's, for example, as a representative, the pyloric rhythm is AB, and we knew from Andy Swenson's work and with Eve that captor and proctolin excite AB. We know that we didn't know what was going on in the gastric mill circuit. And so the gastric mill circuit is over here, simplified, of course. I'm only showing you the LG and its reciprocal inhibitory partner, interneuron 1. And those were the first targets of our studies. And so the question was, are all transmitters targeting the same neurons, or is there divergence in the co-transmitter actions? And so here's an example of LG. Top is an intracellular recording of LG, while we inhibited some other neurons to make sure that it's specific to LG. We apply CAPTERP focally to the STG, and you see a nice excitatory response in the LG neuron. So it responds nicely to CAPTERP. We could not find a response to proctolin. So clearly there's already some divergence going on. Here's interneuron 1. It responds beautifully to GABA in an excitatory fashion. So that was a bit unusual, but it did not respond to the other peptides, to the peptides. And so there's, again, divergence happening here. The interesting, we did a lot of blocking experiments to make sure that this is actually the case, that we're not missing something here. But the thing that blew me away when we did these experiments was that LG has receptors for GABA. So when you apply GABA to the STG and you record LG's membrane potential, you see an inhibition in LG. That happens in LG. So it has receptors for GABA. But blocking these receptors makes no difference for how LG is excited by MCN1, meaning that the GABA that comes from MCN1 does not affect LG. It goes elsewhere. Right? So there's lots of divergence going on here with the transmitters. And I'm showing you just, we had this large map of all of the gastric mill neurons in there. I'm highlighting you the LG neuron. On the left side is the response to focal application. It responds to captor, which is this side. It responds to GABA, which is this side. It does not respond to proctolin. On the right side is the response to neuronal release from MCN1. It responds only to captor. So clear divergence of co-transmitter actions. There are other neurons that respond to all of them, right? At least when you focally apply them, not necessarily when you look at the release from neurons. And so one of the big sort of take-home messages was that there's BAF application of transmitters is definitely not the same as neuronal release of transmitters. Okay, so at this point, I want to address the elephant in the room, that all of these things that we have done that I've shown you so far, for the most part, were done in an isolated nervous system preparation. That had always bugged me a little bit. And so in my lab in Germany, we started to work a little bit on in vivo recordings. Those are difficult, but I think as far as they go, I think what we need to know is, do the neurons that we talk about all the time, that we record all the time, do they actually respond similarly in an intact animal? And so here's again, Uli and this is Florian, who was mentioned by Mikey earlier on, taking this on. Uli did recordings in the intact animal. Again, she recorded from the MCN1 neuron on the ION. This is an in vivo recording on the bottom there. You see MCN1 is nicely bursting here while the pyloric rhythm is going on in the intact animal. This looks very much like in an isolated preparation, so excellent. There's also a correlation between the MCN1 firing frequency and the pyloric cycle period, which is what we would expect from the isolated preparation. If you lesion the ION and you cut essentially the axon of MCN1, you see a drop in pyloric cycle frequency, or if you plot it as period, it goes up in period. It gets slower because you're removing modulation that's ongoing in the intact animal. You can stimulate the ION and you get a gastric nerve rhythm, like in the isolated nervous system. And when you apply chemosensory stimulation, right, to the antennae, we know that the gastric nerve rhythm can start. And in this recording, this is actually the same experiment that I've shown you earlier, where you saw the IV neurons. Only on top of that also recorded the ION. And what you found is that MC1 is more activated with the chemosensory stimulation, as we would expect. So good, there was no problem. We were very happy with this result. Of course, it was essentially just reproduction in the intact animal. This brings me quickly to Florian. Mikey has already talked about this, so I'm not going to go into the detail. But one of the questions was, does sensory stimulation in an intact animal elicit distinct motor patterns as well? And Florian took on this task by recording, different motor nerves, by stimulating different sensory pathways, either electrically or, with a little stick that he stuck down the stomach of the animal while he had an endoscope in there. And what you see here is the tooth movement. This is the right lateral tooth that moves as a motor pattern is on. Here's the gastric nerve rhythm. There's the LG neuron. Whenever LG is active, we have a protraction in the lateral tooth, as expected, of course. And then there's a retraction as soon as the LG burst stops. And then slowly we have a buildup again. And here's also the DG neuron firing. Main point here is, depending on which sensory neuron you stimulate, you get a very different gastric nerve rhythm out of it that you can clearly identify. And so here's an IV neuron stimulation elicited the gastric nerve rhythm. You see the movement of the lateral tooth, the movement of the medial tooth. Here's a POC pathway stimulated gastric nerve rhythm. So there's a very different pathway that I haven't really talked about. Also, shout out to Dawn, of course. And then VCN, this is the gastric nerve rhythm. The only thing you need to see about this is that they all look different, right? That was the whole point of that. So even in an intact animal, we see differences. It's not just on the motor pattern side. Those motor patterns, the different motor patterns translate to the behavior. All right. So going back to our little schematic, right? Is there feedback to, that was sort of like, we could find that the things that we saw in an intact animal, in an isolated preparation, was also true in the intact animal. Now, let me go back to my own passion here, sensory responses, right? And so another question I wanted to ask is there feedback to the sensory system? Is there feedback, for example, from those modulatory projection neurons onto the sensory pathways themselves? So as the sensory information come in, can our central nervous system determine what information we receive? And that brings us to another topic that we have worked on quite a bit, and that's ectopic spike initiation. So let me quickly tell you what that is. So ectopic spikes are generated in the axon, not at the typical spike initiation site, not at the axon initial segment, for example. So here you see a neuron that has its soma up here with dendrites, and then an axon here. And ectopic spikes would be elicited somewhere along the axon, but they can travel in both directions. So antidromically towards the soma, and in the right direction forwards towards the dendrites of this neuron. Now, that is an interesting concept. It has also been shown in many sensory neurons and in other systems, not just the SDG. Of course, we knew that it was true in SDG because we had seen this in the PD neuron, in the LG neuron in the mid-90s. But in like dorsal root ganglion cells, we also find this. And in that case, in those sensory neurons, it was associated with chronic pain. And so in the SDG, we have a neuron that shows ectopic spike initiation. That's a sensory neuron called the AGR neuron. This is approximately what this neuron looks like. Here you see a staining of this neuron. This is a muscle tendon organ that has a soma somewhere in the SDG, and then two axons, one below and one above this, the soma. And it can be investigated with respect to the ectopic spike initiation. So the first thing that we did is we're trying to find where is this spike initiation zone, right? Where can we find it? And so are there specific axonal regions that facilitate this ectopic spike initiation? And if so, is that site of ectopic spike initiation actually functionally important, or is this just an artifact, right? Is this like a thing that happens because we dissect it out or whatever? So how do you find the spike initiation site? That was not easy. And so we tried a lot of things, and the thing that we actually stuck with was quite nice. So we have I told you about optical imaging before. We have a lot of tools available. We have a dye that stains axons quite nicely. And we can activate activity in these axons by just illuminating the axon with blue light. I don't know why it's green in this case, but it should be blue light. And so what you can do is you can record the axon on two sides. You can move your illumination site. And as you pass the original spike initiation site, you can measure the timing between the two recording sites and find out that this is where the original spike was. But just measuring timing differences. And so when you do that, you see something like this. So we move the stimulation, or we move the excitation down the axon, and our spikes move forward in time. And the red one is the one that matched the original spontaneous activity. And so we knew this is now where our spike initiation site is, and we could locate it to approximately 250 micrometers at the border of the SDG. So a little bit above, a little bit below the border of the SDG is where the spike initiation site is. The nice thing about this, it has a fairly high spatial resolution, right? Again, 250 micrometers plus minus is what we were able to do with this. So, okay, so there is one site that is important. Now, is this an important site as well for the neuron? Well, at that location, it turns out AGR has actually little, little neurites that come out of the axon, and we have found that almost all of the AGR axons that we stained had a little neurite exactly where the border of the SDG is. And so that site, of course, we know is a site where lots of interactions happen synaptically. And so we were curious whether we could find synaptic inputs, or inputs that would change the ectopic spiking of AGR. And sure enough, we found two influences. One from the IV neurons that I've mentioned before, the chemosensory pathway that comes down from the brain. There's a slow inhibitory effect on the spike initiation site here. And we find a fast excitatory effect from some of the motor neurons, the GM motor neurons. So let me show you what that looks like. So here we have an IV neuron stimulation. This is our firing frequency in AGR, the ectopic spiking in AGR. So no sensory spikes here, just ectopic spiking. And we see that there's about 40% drop in the spike activity of AGR over a very long time period, right? This is 100 seconds time scale here. At the same time, we found when we activate the GM neurons, we see that the firing of AGR goes up. But every time when we stimulate the GM neurons, this is a faster effect. So the time scale here is 20 seconds. So this happens almost immediately. So it's a more direct effect. It's also a smaller quantitative effect. So it's about 25% or so of an increase that we see. Now, so yes, there is an effect. So it seems to be specific to this site. We actually know if we move the spike initiation away from this, there's also no response anymore to these inputs. But we do see an input here that modulates the activity of this ectopic spiking in the sensory neuron. So now the question was, of course, do these changes in ectopic firing alter the sensory response? And you can look in two directions. You can look forward, because those ectopic spikes can move, orthodromically and towards the dendrites, or they can move antidromically. So here's the forward part. This is done. This was done by Nelly when she was still in my lab. Here shown with her identical twin, and Farzan, who was also a part of that study in 2009. And what Nelly had shown is that if you change the activity of the ectopic spikes of AGR just by a few hertz, like from 1 hertz to 4 hertz, for example, you see a clear change in the motor pattern that comes out of it. So here, the gastric meridian here is much, much slower on the left side than on the right side. And again, the difference is here, just stimulation of ectopic spikes in AGR going from 1 hertz to 3 hertz. And you can quantify this. Of course, the biggest difference is always between 1 and 3 hertz firing frequency for the AGR. Okay, so changes in that AGR activity were important. What about the other side? Going towards the periphery. And this is something that Maggie de Meet did in my lab. And so she first was interested in, can this, since these action potentials travel towards the periphery where the sensor information is encoded, do they actually get there? First question. And second, do they affect what's happening out there? And so she did optical recordings from the axons down here. This, what you see here, is a desheathed nerve, DG nerve, that innervates the muscle. The muscle is not shown here. It would be where the text is down here. But you see the HER axon like coming in here. And so she did optical recordings down here to show whether the ectopic action potential actually make it all the way out. And yes, they do. You can trace them all the way down towards the muscle, even just before where they innervate the muscle in the tendon there. So that was good. They get out there. So what happens if you then stimulate the sensory neuron? And what you do here is you stretch that muscle because it's a muscle tendon organ. So apply stretch to the muscle, or tension to the muscle, I should say, and compare, the response of HER with these ectopic spikes and without them, essentially. And this is what she did here. So this is at 7 Hertz ectopic firing frequency, 5 and 3 Hertz. And the sensory response when you stretch the muscle is shown in blue. And as you can see, that sensory response is modulated with the amount or the number of ectopic spikes that come in. And you can quantify that, of course, so you get a different sensory burst duration and different the number of spikes per burst out of it when you stretch the muscle, just because ectopic spikes invade that sensory location there. So is there feedback to the sensory system? Yes, absolutely. Modulatory projection neurons and motor neurons feed back to the sensory neurons, although in an unusual way on the ectopic spikes in that case, and they alter the sensory response. All right, so that was a lot about sensory stuff. Now I want to switch gears a little bit and talk about what the effects of modulatory neurons are on interneuron and motor neuron activity. And that actually goes back to, something that has bugged me all the time. I started out in stick insect, like I mentioned. My first project there was recording intracellularly from inter and motor neurons as you touch the animal. So here you see a sensory stimulus. Down here, there was a leg movement. You see a recording of a motor neuron. You see it responds to our stimulus. And then I touch the animal. And what happens there is that for a short amount of time, your response to the sensory stimulation is much higher. This is just for like 10, 20 seconds or so, but it's nicely quantifiable. You can also record this in all other kinds of motor neurons and interneurons in there. And this is just an example here. Here is a fast motor neuron before the touch and after the touch, and its amplitude or its response is much stronger. And we tried everything at the time to figure out how the hell does this work? How do we get a stronger response, from a sensory input without changing anything in the sensory neurons? We couldn't find a reason why this would work. And these days, I like to think of this as a modulatory effect, that you excite the whole system as in terms of neuromodulation. And so the question essentially was, how can a motor system go from less excited to more excited without obvious changes in its sensory inputs, right? And this is where the modulation comes in, I think. Now, the story that I'm going to tell you is a story that goes back 21 years, so be prepared for that. It starts, as so often, with Mikey. Here he is in his office, about 2001. It goes to Steffi Heigele in 2007, then to Carola in 2010, and then to Maggie DeMate in 2020. So that's how long we worked on this project, and we still haven't fully understood it. So what was the problem? Well, I was in Mikey's office. I was doing experiments where we looked at how far peptide transmitters actually can spread inside of the ganglion, and was using substances called phosphoramidin and spantide, who are, determining how far peptides can spread in the ganglion. And so I was applying these substances by hand, like I had a well around the STG. I was putting them in manually with a syringe while I was recording from the LG neuron, and I had a gastric nerve rhythm going because I stimulated the MCN1 neuron. And the challenge of these experiments was, first, shaky hands. Two, that you don't lose your recording while you do this. And I got a nice response in a lot of my experiments initially. The problem with this was that I was able to reproduce that response by just applying saline. And so what I was doing wrong here was that the temperature of what I was applying to the SDG was not matched to the actual bath temperature of the SDG. And so whenever I applied any of these substances, I changed the temperature of the SDG, and that caused the major effect that I was measuring at the beginning. Of course, later on we did this right, just to be clear. But at the beginning, we struggled with this. And so this led us to study temperature effects. And so we could reproduce this by just cooling the SDG at 10 degrees Celsius, get a nice gastric myrmeidom. At 13 degrees Celsius, it fails. At 10 degrees, you get your rhythm back. So it's not a permanent damage that you're exerting here. You can repeat this. And so over the time we studied this, then Steffi Heigle in my lab in Germany recorded all kinds of parameters that change with temperature. The membrane potential hyperpolarizes, the spike amplitude gets smaller, the PSPs that come in from MC1 get smaller. And so all that shouted at us that there's an increase in leak current, an increase in the conductance that shunts our membrane. And so we measured some of this. We measured the currents, and as expected, when you increase the temperature, you get an increase in conductance here, increase in leak conductance, because it's mostly linear. It's not fully linear. So there are other effects, obviously, but part of this is the leak conductance that increases. And so we asked the question, can this leak conductance be sufficient to cause the temperature-induced termination of the gastric myrmeidom that we saw? And so we did a dynamic clamp experiment where we introduced additional leak, and in particular, the amount of leak that we measured in our experiment. So for each animal, we measured how much leak was introduced by changing the temperature, and then took that and did a dynamic clamp and introduced the same leak, and then looked whether this was sufficient to crash the temperature. And here's the experiment: 10 degrees, nice gastric myrmeidom, dynamic clamp, adding the leak, and no more gastric myrmeidom. On the flip side, if you now just increase the temperature, no dynamic clamp, the rhythm crashes as well. And now if you subtract that leak, you get your rhythm back. So this was a clear indication that an increase in leak is responsible for the crash of the gastric myrmeidom at those elevated temperatures. Now, what does that leak do? Well, you would expect it to shunt the membrane, right? So you get a reduced length constant, a reduced spread of synaptic signals, and that's something that Maggie DeMate did in my lab when she did optical imaging. So this is a calcium imaging of the LG neuron. As you stimulate, like one stimulus at a time, the MCN1 neuron, and what you see here is the calcium spread in the LG neuron at 10 degrees and at 13 degrees. And if you squint and look exactly, you see there's more green and more blue on the left side than there's on the right side, which indicates that the signal spreads less and there's less of an amplitude of that signal. Of course, we knew this because we had recorded the PSPs of MC1 in the LG neuron. But to quantify this, I'm not going to tell you through, pull you through all of this data here. But if you look at this little figure up here, that's the spread of the postsynaptic signal in LG in terms of area, how far it spreads. The blue line here is at 10 degrees, and the red line here is at 13 degrees. So there's a clear, significant difference in how far the signal spreads inside of the LG neuron. And you can imagine if it doesn't spread as far, you struggle to get to spike threshold, which is shunting the membrane. You can't get to spike threshold, and you fail your action potentials. So with that, we knew that in an intact animal, remember, do the things that we do in the isolate preparation actually matter in an intact animal? That was one of my questions earlier. In the intact animal, that didn't seem to be a problem. We could put an animal to 10 degrees, 13 degrees. You could put it at 18 degrees, and we would see a gastric myrmeidom that was continuously going on in these animals. And in fact, there was almost no dependence of the LG spike activity on the temperature in an intact animal, which was in stark contrast to what we saw in the isolated animal, isolated nervous system. So we were asking what could be the mechanisms, how this is compensated in the intact animal. And so the first hint was that, with higher MCN1 stimulation, we could actually broaden the temperature range at which we could maintain our gastric myrmeidom. And so the other thing is that we found that MCN1 increases its activity when you increase the temperature. This is an isolated nervous system, comes with a lot of caveats, this experiment. But when you look at this here on the left side, MCN1 is the larger unit on this recording. At cold temperatures and at warm temperatures, you see that the MC1 activity goes up. That is reliably so. It's about a 50% increase in MC1 activity. Again, lots of caveats with this experiment. I understand that. But can this increase in MC1 firing frequency rescue our gastric myrmeidom? Sure can. If you have it at 7 Hertz stimulation, you get a gastric myrmeidom at 10 degrees. 13 degrees, it fails, and then you slowly increase your stimulation frequency and you get a gastric nerve rhythm back. So that worked. Now, why is this happening? What's going on? We know, of course, that MCN1 releases CAP-TERP, right? I've shown you this before. It's the only transmitter from MCN1 that affects LG. And so, of course, we thought, well, this could be more CAP-TERP being released. And so, in fact, when you apply CAP-TERP, you can rescue your gastric nerve rhythm from the heat effects. And so here's our gastric nerve rhythm at 10 degrees. Here's the failure at 13 degrees. Here's applying CAP-TERP at 13 degrees, but not stimulating the MCN1 rhythm. So you're not getting a rhythm here. So CAP-TERP by itself does not create a rhythm. But if you then match it with stimulation of the MCN1, you get a nice gastric nerve rhythm back. And of course, you can wash this thing out. And this is reliably so. So you can actually do this with any peptide that causes the same current in LG, the IMI, the modulator-induced current, as long as LG has receptors for it. And so here's a CCAP experiment, crustacean cardioactive peptide. Same idea, 10 degrees, you get a nice gastric nerve rhythm. 13 degrees, you crash the rhythm. Then you add CCAP to the bath, no gastric nerve rhythm. You stimulate on top of that at 13 degrees, you get a nice gastric nerve rhythm back, and you can wash that out. So what is actually going on here? Well, of course, our idea is that IMI can act as a negative leak conductancy. And this was a suggestion that came out in like 2010 by Sheng Bing in Farzan's lab. And when we heard that at around that time, we were like, oh my God, this is the solution to our problem. This is what we couldn't understand at the time. And it really was an eye-opener when I talked to Sheng Bing at the time about IMI. And so can IMI by itself counterbalance, the temperature effects? So it can. So we did an dynamic clamp experiment here. We had our LG activity here. This is the rhythmic activity of LG in an MCN1 gastric nerve rhythm. We crash it at 13 degrees, and then we introduce IMI, not the peptide, just the IMI into LG, and we see that the rhythm can be rescued at a conductance level that LG actually sees, right? This goes back to something that Mikey has done with Nick DeLong a while ago, where they measured approximately what the strength of IMI is in LG. And so that was nice. So our summary of this is that gastric nerve rhythms terminate at elevated temperature. The termination is caused by the increase in leak conductance, and that increase in leak can be balanced by peptide or IMI neuromodulation, right? So now on to a little more today's stuff, right? We know that this is not only true for the MCN1 gastric nerve rhythm, and so this was a more recent study that we did where we had a VCN rhythm that is a long-lasting gastric nerve rhythm, right? The mechanosensory neurons that I mentioned earlier elicit this. You see our nice gastric nerve rhythm here. This is at 16 degrees at the STG. The rest of the bath we kept cold for a reason. I'll get to that in a second. And we see that at this temperature we see nice gastric nerve rhythms in all animals that we test. Now, if we then increase our temperature by just 3 degrees, right, to 19 Celsius in this case, the gastric nerve rhythm crashed, and this was consistent across animals as well. Now, the interesting part about this is if you now heat the whole thing, now you're heating the COGs along with the STG, you can rescue the gastric nerve rhythm. So here at 19 degrees for the whole bath, including the projection neurons and the upstream ganglia, right? And everything that's in there, including axons and everything. You are now rescuing that rhythm from its heat effects. So heating the upstream ganglia that contain the projection neurons rescues the gastric nerve rhythm and provides some temperature robustness. And so for us, this is a clear indication that increased neuropeptide modulation is involved in temperature robustness, right, to the gastric nerve rhythm at least. And so I think this is a pretty nice result. And this is where we are with this project currently. And so now I'm looking at the time and slowly getting to the end here. I promised, I wanted originally to put in some of our work on migraines and so forth. But I at least wanted to give you a little teaser, a little cliffhanger about what I've done in the last year. In the last year, I've been looking at comparative approaches, studying the impact of climate change, temperature changes in the ocean, and neuropeptide actions in other species of crustaceans, decapods, obviously. And so I've studied invasive species, and I studied some local species, and I compared their temperature responses to one another. This requires a bit of more time than I have left here, but I would be happy to talk to you about this. Maybe Dawn, at some point, maybe I've changed my mind about the SDG meeting. Maybe I can present this at the SDG meeting instead. I looked at the data. I think I could present this. Anyways, but a little cliffhanger. I found something that is very strange to me, and I'm very interested in what you are about to say to this. So here is a recording of the pyloric rhythm of this little critter here called Hemigrapsus tecanoe. That's the Japanese shore crab or Asian mud crab. That is an invasive species in Europe. It's small, it's just like the size of a quarter. So I had some challenges with dissecting this critter. But I was able to record its temperature responses. This is pyloric rhythm, obviously. So let's look at the middle here. Here is like at around 21 degrees Celsius, the PDN, the LVN, you see the nice pyloric rhythm that's going on here. It has a period of about 2 seconds. Now, the interesting thing that I found is, first of all, when you go colder, which is upwards, the cycle period goes longer. So the way I plot this is a little weird here, but at like 5 degrees Celsius, the cycle period is about 3 seconds. The fun fact about this is if you get warmer, it also gets slower. And so at like 27 degrees plus, I found like 4 seconds of a cycle period. So this thing has an optimum where it's the fastest, and it gets slower when you get colder, and it gets slower when you get warmer. And even more to that, the phase relationships change as you do that. So at cold temperatures, PD has one of the longest phases. The PY phase is relatively short. And as you go to warm temperatures, you get this long PY phase and this relatively short LP phase, which is clearly different from, the room temperature at 21 degrees Celsius or the cold temperatures. More than that, though, it's independent of the cycle period, because cycle period is slow at cold and slow at warm, but fast in the middle. So it doesn't depend on cycle period. So with that, this is just a teaser. I certainly have to go back and analyze the data, but I can certainly show you more of that data if I had more time. For now, I want to stop here. Those are the things I didn't get to talk about. Unfortunately, there's the microgrid models that I didn't have time to talk about, and more about the climate change stuff. A little bit more about like crayfish genomics that we have done. But for now, let me just thank the people that have contributed in one way or another to these studies. And with that, I'm done. I'm happy to talk to you and to answer any questions that you might have.
I have a question, Wolfgang.
Go for it.
I don't know what the current state of all the potential imaging stuff is in our system, but I was wondering if there's a possibility that it's gotten to the point where you'd be able to start tracking which projection neurons or even how many projection neurons are co-activated under a given condition.
Yeah, I think this was one of the things that we tried to do. A problem with this was finding which one is the projection neurons that we are looking for in the sea of neurons that is up there. We know where approximately they are, but the finding them is always in retrospect, right? So you do the experiment, you image the neurons. You can definitely tell which ones are activated, which ones are not activated. That's not a problem. But then finding which neuron is which, you have to essentially stain them afterwards or do an intercellular recording from them and try to find them afterwards. So that was the part that we struggled with at the time. And my PhD student and my was at that time not able to do that experiment essentially. So unfortunately, that was one of the goals that we had, never got done. But in principle, it's doable. It's difficult to do though. The quality of the recording, of the optical recording, would allow you to do this.
I see. That's not the limitation.
That's not the limitation there. The signal to noise ratio is not the greatest with these voltage sensitive dyes, that's for sure. Calcium dyes don't work. I've tried quite a few. They just don't work well if you apply them extracellularly. If you inject them, fine. But bath application, not so much. We miss the esterase, right? That activates them.
Well, keep at it.
I need people. Since I've been gone for a year, my lab is essentially empty. I have currently one new master student and another student that I share who works on like crayfish stuff, but not SDG stuff. So I currently have no one really intensely working on SDG stuff.
Other questions?
Wolfgang, going back to AGR and GM1, can you talk a little bit about the mechanism? So I can imagine you might have, collision, but I'm trying to think about how a lower frequency, if the action potential is heading to the muscle, is shaping the output from the tendon organ.
Yes, we thought about this quite a bit. We have no experimental evidence how this works, but we think that there is a slow conductance, some slow potassium conductance that's out there where you, when you stretch the muscle or put tension on the muscle, that is contributing to the sensory response, to the sensory burst that you're creating there. And if you have more antithrombic action potentials that come in, they also sum up there, and that is affecting how strong your burst is in the periphery. And we've seen that with more ectopic action potential, your peripheral burst gets weaker, right? And that fits that view. But again, we have no experimental data. Wouldn't know how. We thought a long time about how to do this, but couldn't come up with a good experiment for it. It is not the collisions, just to be clear. Like, we have made sure that in our experiments, the collisions were excluded from that analysis. So, we used frequencies where we sort of like avoided any collisions that were happening in the system. If you do have collisions, very clear, then of course the spikes that you get from the periphery don't make it to, along the axons and they collide. However, Nelly in her works in 2009 has already shown that there are very few collisions. So essentially you collide one spike, and then because of the higher spike frequencies from the periphery, all the other spikes are sensory spikes. So there's really just one collision that happens on the length of the whole axon, and all the other spikes will not be impacted by the ectopic spiking. So it's really, they're getting out in the periphery and doing something there.
Okay, thanks.
Yeah, Omar.
Yeah, thanks for the talk. So I have two questions. One is when you showed that the difference between response in bath application versus neuronal input of modulator, right? Was that how? So what was the metric for response? Did you measure current or behavior or like activity? And
yes,
so
we measured whether the neuron responded to our bath application in that case. So it was a focal application to the SDG neuropil. So there was a puff electrode that applied the peptide or the GABA to the SDG neuropil, and we recorded from the neuron and saw whether the neuron was depolarized or hyperpolarized. So we didn't characterize any receptors at the time, and now we of course can go back and look for receptors with transcriptomics. But at the time we did this, we took care that we didn't miss anything, because some of these effects that the peptides elicit are voltage dependent. So you might sit at a negative voltage and you wouldn't see a response, but if you were depolarized, you would see a response. And so we did that. We changed the membrane potential as well and looked for responses.
Okay, in any given cell type. But I mean, like, if so, did you guys do like synaptically isolate?
Yes, that's the other thing that we did. We did these. Yes,
okay.
We did this in low calcium saline partly, or by blocking the other inputs, or dual recording from other neurons that innervate those neurons, so that we were sure that all the synaptic inputs, or at least most of them, were suppressed. And so there was a lot of like dual recordings. And of course, we know which neurons innervate which neurons in the system. And so we can hyperpolarize the input neurons to get rid of synaptic inputs, even without synaptic blockers. But yes, some of these were done with synaptic blockers as well.
Okay. Yeah, I was just curious if that was part of the discrepancy, if it was like some sort of synaptic.
Oh, that's absolutely, you're right. This is why we did this the way we did it, right? It was a lot. I glossed over this, right? Since Mikey had talked a little bit about this, and there was a lot of experiments involved where we did exactly this, making sure that it is not an indirect effect that we are measuring. I would still feel, more confident in that if we went back and looked at receptors at this point. And
Sure,
I'm sure
that's
this
fair.
is somewhere in the
Okay, cool.
transcriptome.
Yeah. All right, cool, thanks. So the second question is when you're looking at the gastric mill rhythm and the change in temperature and how cooling it down basically kills the rhythm, and you can recover it by adding negative leak, right? That was
Yes,
one of the
it's
results.
heating the rhythm kills it.
Heating it up.
Yeah.
Heating it up. So,
Yes.
and the conclusion there is that leak is involved somehow, right? And is it fair to say that it might not necessarily be the leak current itself, and just, I mean, it's hard to say that maybe it isn't like some decrease in like calcium conductance, for example, right? So some net effect on excitability that you can recover, right?
Yeah, no, absolutely. I mean, we saw that a linear leak increased and that it was sufficient to rescue the rhythm. Let's put it this way. There is, of course, no problem, no discussion about that other currents increase, with temperature. And of course, that would contribute to the total conductance increase naturally. Like any current will increase with its own temperature response, with its own Q10 value. And so as you increase the temperature, we'll get more conductance increase in most of the currents until you get to, it becomes a little bit more difficult if you talk about sodium currents that have activation, inactivation gates. But generally, you get more conductance in all of your currents. And some of them will, of course, contribute to that temperature response and also to the failing. I mean, when you think about the pyloric rhythm, that seems to be different, because in that case, as you increase the temperature, first of all, it's more temperature robust per se than the gastric rhythm. Second of all, once you fail, I think it's mostly related to, some currents increasing more strongly with temperature than others, and you're losing that balance between the different currents, and then you fail your activity. This has probably also to do with how the two different rhythms are generated. One is more based on network activity and synaptic inputs. The other one more on intrinsic, first generation in AB as a pattern generator. And so, but you're absolutely right. I'm not claiming that there's no other effects on this. I'm just saying that the leak plays a big part in this, and that IMI is ideal to counterbalance that leak increase.
Cool. Appreciate it. Thank you.
Yeah.
Wolfgang, I want to return for a moment to my previous question to ask, you cannot identify yet the projection neurons without some intensive after imaging work. What you presumably can do from the imaging alone is determine what subset of those active neurons are exhibiting a pyloric and or gastric mill-like pattern.
Yes.
So was it most of them, some of them, few of them?
Yeah, I don't remember.
Or don't you remember?
I know that the ones that I've shown you were all involved in gastric mill patterns activity. So there's a lot of them that respond to gastric mill patterns, because those are easy to identify in the voltage signal, in the optical signal, because they're slow changes and we can average one. I do remember seeing a lot of them having pyloric activity, but I cannot tell you, which ones have both and what the numbers are. I would have to look again.
But at any rate, it sounds like it was more than one or two.
Yes. Yeah, definitely more than one or two. That's for sure. There's a lot going on in these neurons, made our life more complicated. What I really would like, this is something that we've been trying to do for a long time. We are working on these crayfish. We have the genome in this crayfish. We have some collaborators in Germany that have done RNAi in there, and we have done some RNAi, but also we've been trying to genetically modify these animals for a while now with a technique called remote that was by Jason Rascon, invented by Jason Rascon at Penn State. And we are able to transport in CRISPR constructs into the oocytes of these animals. And one of the first things that I want to do when this finally happens, it's been a long time, I want to express a calcium indicator in essentially all neurons, a pan-neuronal calcium expression. And if I take this, I can throw the COGs of a crayfish under the microscope and can monitor all of these neurons at the same time, essentially, and hopefully answer that question more easily without all the voltage sensitive dye stuff and the low,
Right.
the low signal to noise ratios. So that would be nice. Send me someone to help with this.
That'll be good. Marie, you're being photobombed. All right, any other questions or comments? Looks like the end of another monthly meeting. Thank you all for coming. Thanks, Wolfgang, for a great talk.
Yeah, thank you. It was nice. See you all.
See you all. See you in San Diego.
Bye.
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 Bye.